I'm Yerem Yeghiazarians. I'm the co-founder and CEO of Soley Therapeutics. So I think the key here when you're trying to describe this to people is to try to, again, differentiate the approaches. And why do we think our approach, hopefully, to be determined, frankly, it has to be done in clinical trials, we think has a great advantage. So traditionally, what's done is, again, as I said, people come up with a disease of interest, and then they study for years and decades what is the etiology of that disease to come up with a target. And then they come up with a drug that binds that target. They do a lot of medchem, and there's a lot of AI/ML involved with medicinal chemistry these days to bind the target better, have predictive ways of binding the target better, and then they have to validate it. They do all the downstream experiments. Well, that's one way of doing it, and at times it works. But the incorrect target selection surfaces late in development, right? You think you know the target, but that target may be the wrong target. It may be insufficient target, and you won't know that until the clinical trial fails. And unfortunately, for complex diseases, as people and you yourself are well aware of, there's over 90-95% failure rate for many reasons. But this is one of the main reasons, because we unfortunately, despite advances in science on many fronts, we don't know the target to go after for many of these diseases. So we decided not to do it that way. So our approach initially identifies novel molecules that drive a desired cell fate. That's really important because we can filter out broadly toxic molecules early. That's important. Number two, once we have a drug that gives rise to those phenotypes, we can define key targets, networks, and mechanism of action at the whole cell level, not just the one upstream target. And this allows us to uncover novel targets and mechanisms that otherwise would have been missed. So that's another important point. Notice that I still haven't told you about disease and patient selection. We essentially have now a drug that we are very high on. It passes druggability criteria. It's been filtered out the toxicity, and we have novel MOA. It's only then that we've put a completely novel approach to figure out which disease and which patients with that disease should then enroll in a clinical trial. That's what I meant by flipping the whole thing 180 degrees. We don't start with disease, target, drug, downstream experiment. We flipped it around. We discover the molecule at the cell level, do the medchem, figure out how it works, then figure out which disease and which patients to go after. So this provides an unbiased view from the start, and the cell is helping us make the best selection.